Hello students, today I bring for you new chapter Biotechnology Principles and Processes. In this chapter we are going to solve a total of 45 questions which are coming the previous 10 year of exam, that is from 2013 to 2023. So let's start the first question. Ligation of foreign DNA at which of the following site will result in loss of tetracycline resistance of PBr322? Option A PST1, option B PVU1, option C ECOR1, option D BAM-H1. So here the answer will be option D BAM-H1. The BAM-H1 site is within the tetracycline resistance gene. If foreign DNA is inserted at this site, it would disrupt the tetracycline resistance gene, causing a loss of tetracycline resistance. So D option is the correct answer. Question number second. Which of the following is not a cloning vector? Not a cloning vector, okay? So YAC PBR322, probe BAC. Here the answer is option number C, probe. Okay? C is the correct answer because a single stranded DNA or RNA tagged with a radioactive molecule is called a probe, and it helps in the detection of mutate gene. So C option is the correct answer. Question number third. Two statements are given, you can see. Assertion region type questions. Assertion. When a particular restriction enzyme cuts strand of DNA, overhanging stretches or sticky ends are formed. Okay. Correct one. Reason. Some restriction enzymes cut the strand of DNA a little away from the center of palindromic site. Reason also correct. So here the answer will be, according to the options, the answer will be option number B. Both A and R are correct, and R is the correct explanation of A. Next is question number 4. Separation of DNA fragment is done by a technique known as gel electrophoresis, PCR, recombinant technology, southern blotting. So here the answer will be option number A, gel electrophoresis. Okay. Separation of DNA fragments which is carried out after restriction enzyme digestion is done by a technique known as gel electrophoresis. Next question number 5. The enzyme A is needed for isolating genetic material from plant cell and enzyme B for isolating genetic material from fungus. Choose the correct pair of the options from the following. Enzyme A और enzyme B क्या है? Enzyme A is cellulase and enzyme B is chitinase. Option C is the answer. Okay, plants material के लिए cellulase और फंगस मटेरियल के लिए काइटिनेज. सो ऑप्शन सी आपका आंसर होगा. नेक्स्ट वन, क्वेश्चन नंबर 6. मैचिंग दिया गया, देखो. जीन गन, जीन थेरापी, जीन क्लोनिंग, जीनोम और लिस्ट 2 में उनके बारे में डिटेल्स दिया गया है. सो जीन गन इज कनेक्टेड टू option number 2, used for transfer of gene, and gene therapy is the replacement of a faulty gene by a normal healthy gene. 1 point में आ गया पहले में. Then gene cloning is to obtain identical copies of a particular DNA molecule, and genome is the total DNA in the cell of an organism. So according to this, option number 2nd, 1st, 4th and 3rd. 2nd, 1st, 4th and 3rd. Option number B will be your answer. Explanation. Gene gun or biologistic is a direct gene transfer method suitable for plant cell. Gene therapy involves replacement of a faulty gene by a normal healthy gene. Gene cloning is done to make identical copies of a particular DNA molecules, and genome is the total DNA in the cell of an organism. So option B your correct answer. Question number 7. Pathogenic bacteria gain resistance to antibiotics due to change in their nucleoid, cosmids, plasmids, nucleus. So here the answer is option C, plasmids. Explanation: plasmid is small circular DNA outside the genomic DNA. The plasmid DNA confers certain unique phenotypic characters to the bacteria such as resistance to antibiotics. So option C is your correct answer, plasmid. Question number 8. Which of the following method is not commonly used for introducing foreign DNA into the plant cell? Not commonly used, okay? Not दिया गया है. Sorry. Bacteriophages, agrobacteria medicated transformation, gene gun, and option D, disarmed pathogen vectors. Which method is commonly not used for introduction of foreign DNA into the plant cell? So here the answer will be option A, bacteriophages. Option A is the correct one because bacteriophages act as a cloning vector for bacterial cell, not the plant cell. So A option is the correct answer. Question number 9. Refer to the following statement for agrogel electrophoresis और यहाँ पर आपको चूज करना है appropriate आंसर. तो देखो, agrogel electrophoresis. Agrose is a natural polymer obtained from cuet. Okay, correct है ये regarding agrogel electrophoresis. Option B, the separation of DNA molecule in agrogel electrophoresis depends on the size of the DNA. ये भी correct है. The DNA migrates from negatively charged electrode to the positively charged electrode. Negative से positive की तरफ बढ़ता है, तो C भी correct है. D की तरफ आते हैं. The DNA migrates from positive charged electrode to the negatively charged electrode. It is the wrong one. So we can say option number C, ABC only is your answer. Option C is the correct answer as the most commonly used matrix in gel electrophoresis is agrose, which is a natural polymer extracted from cuets. So option C is your answer. Question number 10. Given below are two statements, assertion and region type questions. Assertion: polymerase chain reaction is used in DNA amplification. Region: the amplicillin resistance gene is used as a selective marker to check transformation. Well, here both statements are correct one, but region is not properly explaining the assertion. That's why option B is your answer. Both the statements are correct, but the given region is not the correct explanation of the assertion. Polymerase chain reaction is used in DNA amplification, and amplicillin resistance gene is a selective marker to help the check of transformation by selection of transformants. Next question number 11. Which one of the following statement is not true regarding gel electrophoresis technique? The process of extraction of separated DNA strand from gel is called elution. यहाँ पर not true पूछा है, ओके, ध्यान देना. पहला स्टेटमेंट क्या दिया? The process of extraction of separated DNA strand from gel is called elution. ये correct है. The separate DNA fragment are stained by using ethidium bromide. This one also correct one. The presence of chromogenic substrate given blue colored DNA band on the gel. This one is the wrong one. Orange color देता है. Bright orange colored band of DNA can be observed in the gel when exposed to UV light. Correct one. So C is the wrong statement regarding gel electrophoresis technique. So option C is your answer. Okay. Option C is the incorrect statement as bright colored bands of DNA can be observed in the gel when ethidium bromide treated DNA is exposed to UV light. Not blue color, it gives bright orange color. Okay. Next one, question number 12. Two statements are given. You can see statement 1. Restriction endonuclease recognize specific sequence to cut DNA known as palindromic nucleotide sequence. Correct one है ये. Statement 2. Restriction endonuclease cut the DNA strand a little away from the center of the palindromic site. This one also correct. So we can say option A is your answer. Both statement 1 and 2 are the correct one. Explanation: option A is the correct answer because both the statements 1 and 2 are correct. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. It will bind to the DNA and cut each of the two strands of double helix at specific point. The restriction endonuclease cut the strand of DNA a little away from the center of the palindrome site, but between the same two bases on the opposite strands. So both the statements 1 and 2 are the correct one. Question number 13. Which of the following is not a desirable feature of cloning vector? Not a desirable feature of cloning vector. Presence of origin of replication, ये तो desirable factor है. Presence of marker gene, this is an also correct one. Presence of single restriction enzyme site, correct. Presence of two or more recognition site, this one is the wrong one. यहाँ पर not पूछा है, so D is your answer. Explanation: option D is the correct answer because cloning vector are the carrier of the desired gene in the host cell. The feature desired in a cloning vector are presence of origin of replication, presence of marker gene, presence of very few, preferably single recognition site for the commonly used restriction enzyme. So option D is your answer. Question number 14. मैचिंग करनी है organism with its use in the biotechnology. Bacillus thuringiensis, Thermus aquaticus, Agrobacterium tumefaciens and Salmonella typhimurium. So here 14 की बात हो रही है. Bacillus thuringiensis, it is a type of cryoproteins. Thermus aquaticus used in the DNA polymerase. Agrobacterium tumefaciens, it is used in the cloning vector, and Salmonella typhimurium, it is the constructions of first rDNA molecule. And according to this, 4, 3, 1 and 2, option number A is your answer. Bacillus thuringiensis is a bacteria that produces cryoproteins. Thermus aquaticus is a bacteria that lives in water and a source of thermostable DNA polymerase that is called Taq polymerase. And Salmonella typhimurium used in the production of first rDNA molecule. Agrobacterium tumefaciens used in cloning vector. Okay, cloning vector. So option A is your answer. Question number 15. In gel electrophoresis, separated DNA fragments can be visualized with the help of. In gel electrophoresis, separated DNA fragments can be visualized with the help of option A, ethylene bromide in UV radiation, acetocarmine in UV radiation, ethylene bromide in infrared radiation, and option D, acetocarmine in bright blue light. So, how can they visualize? Option A is your answer, ethylene bromide in UV radiation. Explanation: DNA fragments are created when restriction endonuclease cleaves the DNA at restriction site. Gel electrophoresis is a technique that can be used to separate these fragments only after dyeing the DNA with a chemical called ethylene bromide and then exposing it to UV light and produce bright orange color band of DNA. So A is the correct answer. 16. The sequence that controls the copy number of the linked DNA in the vector is termed Ori site, palindromic sequence, recognition site, selectable marker. So here the sequence that controls the copy number of the linked DNA in the vector is termed as Ori site, origin of replication. This is a sequence from which replication begins and any fragment of DNA can be forced to replicate within the host cell if it is linked to the sequence. This sequence is also regulates the quantity of copies of linked DNA. So, origin of replication is the answer. Option A. Question number 17. The specific palindromic sequence which is recognized by E-core 1 is specific palindromic sequence recognized by E-core 1. So which one is the answer? Answer is the option number A आपका नहीं होगा, B C T T A H, B भी नहीं होगा, C G G A T C C, C भी नहीं होगा, D is your answer, G A A T T C and C T T A G G. It is the recognition site of the E-core 1. So explanation: each restriction endonuclease recognizes nucleotide sequence in DNA that are palindromic. E-core 1 recognizes the palindromic sequence that is 5 prime, GAATTC 3 prime and 3 prime CTTAAG 5 prime are the distinct palindromic sequence. The sticky ends create hydrogen connection with their complementary counterparts. So D option is the correct answer. Question number 18. Identify the wrong statement with regards to restriction enzymes. Wrong one आपको चूज करना है. देखो, they cut the strand of DNA at palindromic sites. ये correct है. They are useful in genetic engineering. This one also correct. Sticky ends can be joined by restriction enzyme. ये wrong हो गया, तो C आंसर आपका करेक्ट होगा. D, each restriction enzyme functioned by inspecting the length of a DNA sequence correctly. तो ऑप्शन C is your answer. Explanation: sticky ends are joined by DNA ligase enzyme, not by restriction enzymes. DNA ligase seal the end, but restriction enzyme cut the DNA at the restriction sites. Both are used to insert genes and other pieces of DNA into plasmid during DNA cloning. So C is the wrong statement and C is your answer. Question number 19. First discovered restriction endonuclease that always cut DNA molecule at a particular point by recognizing a specific sequence of 6 base pairs is option A adenosine deaminase, option B thermostable DNA polymerase, option C HIND2 and option D E-core 1. So here the answer is option number C HIND2. Explanation: 5 years later the first restriction endonuclease HIND2 was isolated and described. Its functions are dependent on a certain DNA nucleotide sequence. HIND2 always cut DNA molecule at a specific location by recognizing a 6 base pair sequence. And if you asked for the base pair, that is 5 prime GTC GAC 3 prime and 3 prime CAG CTG 5 prime. This is the sequence 6 base pair sequence for HIND2. Question number 20. In a mixture, DNA fragments are separated by restriction digestion, option B electrophoresis, option C PCR, option D bioprocess engineering. DNA fragments are separated by option B jelly electrophoresis. Okay, it is a technique that can separate DNA fragment into mixture based on their sizes. DNA sample are put into a well at one end of the gel and an electric current is used to down the samples through the gel. Negatively charged DNA pieces move towards the positively electrode. So B option is the correct answer. Question number 21. In recombinant DNA technology, antibiotics are used. In recombinant DNA technology, antibiotics are used in option A to detect alien DNA, option B to impart disease resistance to the host plant, option C as selectively marker, and option D to keep medium bacteria free. So here the answer is option C as selectable marker. Explanation: antibiotics are employed as selectively marker in recombinant DNA technology Select will marker aid in the identification and elimination of non-transformant as well as the selectively growth of transformant. So C option is the correct answer. Question number 22 मैचिंग दिया गया है. बायोरिएक्टर इलेक्ट्रोफोरेसिस पीसीआर. आई थिंक एक और लास्ट लाइन ऊपर चला गया होगा. बिल्कुल एलिसा. देखो bioreactors, it connected to option number second because it to the production of large quantities of products. Electrophoresis, it connected to option number 1 separation of DNA fragments. PCR it connected to, I think this option will be the one, amplification of nucleic acids, option number 4, and ELISA connected to option number 3, that is detection of pathogen based on antigen antibody reactions. So according to this, second, first, fourth and third, the answer will be option A. Explanation. Bioreactors that are a large scale production facility for items such as proteins. Electrophoresis, it is a technique that is used to separate DNA fragments based on their size. PCR, polymerase chain reaction, this method is used to multiply nucleic acids. And ELISA, enzyme linked immunosorbent state test, is a pathogen detection method based on the antigen antibody interaction. So option A is your answer. Question number 23. Spooling is. Spooling क्या होता है? Option A, cutting of separated DNA band from the agarose gel. Wrong one. Transfer of separate DNA fragments to synthetic membranes. This one also wrong. Collection of isolated DNA. Correct one. Amplification of the DNA. Wrong one. So C option is the correct answer. Explanation. Using a glass rod or a wooden spooling stick, gently push the ethanol down into the DNA solution during DNA extraction. Because the charged DNA molecule migrate away from the ethanol, they can be scooped up and whirled around a glass rod or a wooden spooling stick. This is referred to as DNA spooling. Long strands of DNA can be isolated from a solution via DNA spooling. So option C is your answer. Question number 24. State the correct statement. Option A. The polymerase enzyme join the gene of interest and the vector DNA. It is the wrong one. Option B. Restriction enzymes digestions are performed by incubating purified DNA molecule with the restriction enzyme of optimum condition. B statement correct है, आपका आंसर भी है. Option C, PCR used for isolation and separation of gene of interest. Wrong one. D, gel electrophoresis is used for amplification of DNA segment. D is also wrong one. So B is your correct answer. Explanation. In order to perform restriction enzyme digestion, pure DNA molecule are incubated with the restriction enzyme under the best condition for the enzyme. To monitor the progress of restriction enzyme digestion, agarose gel electrophoresis is used. The gene of interest and the vector DNA are joined by DNA ligase. The technique of gel electrophoresis is used to isolate and separate gene of interest. And the PCR method is used to amplify a DNA fragment. Okay, so option B is the answer. Next question number 25. Which one of the following equipment is essentially required for growing microbes on a large scale for industrial production of enzymes? Which type of equipment is essentially required? Option A, BOD incubator. Option B, sludge digester. Option C, industrial oven. And option D, bioreactor. So here answer will be option D, bioreactors. Okay, large scale में कुछ भी प्रोड्यूस करना है एंजाइम्स, मोस्टली प्रोटीन्स, तो उसके लिए क्या यूज होगा? ऑप्शन डी, बायोरिएक्टर्स. Bioreactors are essential for the large-scale synthesis of enzymes. Bioreactors are used in the large-scale production. So option D is your answer. Question number 26. DNA precipitation out of a mixture of biomolecules can be achieved by treatment with isopropanol, chilled ethanol, methanol at room temperature, chilled chloroform. Simple. Option is option number B, chilled ethanol. Okay. Chilled ethanol is used to precipitate DNA during the isolation of a targeted gene. In this suspension, this can be viewed as a collection of fine threads. So, B option is the correct answer. Question number 27. Following statements describe the characteristics of enzyme restriction endonuclease. Identify the incorrect statements. So, come to the statements. Here are they. You have to identify the incorrect statement. Okay. Option A. Option A is the enzyme cut DNA molecule at identified position within the DNA. Correct है. The enzyme binds DNA at specific site and cuts only one of the two strands. ये गलत हो गया. दोनों स्ट्रैंड में कट करेगा ना, एक स्ट्रैंड में कट करके क्या होगा. थर्ड, the enzyme cuts the sugar phosphate backbone at specific site on each strand. Correct है. The enzyme recognizes a specific palindromic nucleotide sequence in DNA. Also correct one. So B is your answer. Okay. Explanation. Restriction enzyme recognizes specific sequence and cut DNA molecule at the location. The length of a DNA sequence is checked by each restriction endonuclease. It will connect to the DNA and cut each of the two strands of the double helix at precise spot in their sugar phosphate backbone after found its specific recognition sequence. So option B is your answer. Question number 28. The correct order of step in polymerase chain reaction is. Correct order. First step क्या होता है? Now first step is the denaturation, second step annealing, and third step extension. So option number D is your answer. Explanation. Denaturation of double-stranded DNA is followed by the annealing of short oligonucleotide chain known as primers, and finally the DNA polymerase extends or elongates the newly created strand and amplifying the amount of DNA. So option D is your answer. Question number 29. What is the criterion for DNA fragments movement on agarose gel during gel electrophoresis? Option A. The larger the fragment size, the farther it moves. ये गलत हो गया. स्मॉलर होगा तो ज़्यादा दूर तक जाएगा. The smaller the fragment size, the farther it moves. Correct one. Positively charged fragment move farther end. Wrong one. Negatively charged fragments do not move. Wrong one. So B option is your answer. 29 explanation. The agarose gel's sieving function separates the DNA fragments according to their size. As a result, the smaller the piece, the farther it travels. So B option is the correct answer. Question number 30. The process of separation and purification of expressed protein before marketing is called. Separation purification of expressed protein before marketing is called upstream processing, downstream processing, bioprocessing, and post-production processing. So here the answer will be option number B. Option number B, downstream processing. Okay. Explanation. After the biosynthetic stage, downstream processing involves a variety of activities such as product separation and purification. The product is made with appropriate preservatives. As with pharmaceuticals, such formulations must go through clinical testing. Each product must also undergo stringent quality control testing. So B option is the correct answer. 31. A gene whose expression helps to identify transformed cell is known as. A gene whose expression helps to identify transformed cell is known as. Option A, selectable marker. Option B, vector. Option C, plasmid. Option D, structural gene. So according to the options, the answer will be option A, selectable marker. Explanation: selective marker or marker gene aids in the selection of transformant and the elimination of non-transformants. So option A is your answer. Question number 32. Restriction endonuclease are used in genetic engineering for ligating two DNA molecules. Galat. Used for in vitro DNA synthesis. This one also wrong. Synthesized by bacteria as a part of their defense mechanism. सी ऑप्शन आपका करेक्ट है. डी present in mammalian cell for degradation of DNA when the cell dies. Wrong one. So option C is your answer. Synthesized by bacteria as a part of their defense mechanism. Explanation: restriction endonuclease recognizes cleave foreign DNA in the bacterial cell, killing invading organisms. DNA polymerization enzyme polymerase 3 and Taq polymerase added deoxyribonucleotides to the primer in the 5' to 3' direction. ECOR1 and is an E. coli restriction enzyme that digests DNA at a particular site. The DNA ligase enzyme forms a phosphodiester link between 3' hydroxyl at one end of one fragment and a 5' phosphate at the other end of the another fragment to connect two DNA fragments together. And using DNA ligase, foreign DNA and a plasmid with complementary ends can be ligated. So option C is your answer. Question number 33. Which of the following restriction enzyme produce to blunt end? दो टाइप के एंड प्रोड्यूस होता है. एक तो आपका आ गया स्टिकी एंड, दूसरा आ गया आपका ब्लंट एंड. तो ऑप्शंस देखो, इनमें से कौन ब्लंट एंड प्रोड्यूस करेगा? एक तो आपका कुछ इस तरह के कट करता है, इस तरह से कट होता है, इसको हमने कहा स्टिकी पोर्शन रह जाता है इसमें. और एक आपको सीधा बीच में से कट कर लेता है, इसको हमने कहा ब्लंट एंड कहा गया इससे. ओके, सो यहाँ पर क्या होगा आंसर? विच वन इज द ब्लंट एंड? Which particular enzyme produces blunt end? So here your answer will be ECOR-V. ECOR-5 is a type of restriction enzyme that produces blunt end. Next, question number 34. Which of the following is not a component of downstream processing? Preservation, expression, separation, purification. So here the answer will be option number B. Expression is the answer. Explanation: the procedure includes separation and purification, which are collectively referred to as downstream processing, not the expression. Question number 35. A foreign DNA and plasmid cut by the same restriction endonuclease can be joined to form a recombinant plasmid using. Foreign DNA and plasmid cut by the same RE can be joined to form a recombinant plasmid using polymerase 3, ligase, ECOR1, Taq polymerase. So simple, joining करने का काम किसका है? Option B, ligase. Okay, so this is the answer. Explanation: the resultant DNA fragments have the same kind of sticky end when cut by the same restriction enzyme, and they can be linked together end to end using DNA ligase. So option B is your answer. Question number 36. State tank bioreactors have been designed for. Option A, availability of oxygen throughout the process. Correct है. Option B, ensuring anaerobic conditions in the culture vessel. Purification of product, addition of preservative to the product. So A option is your answer. Explanation: a bioreactor provides the best circumstances for producing the desired product by allowing for optimal growth, that is temperature, pH, substrate, salt, vitamins, oxygens. The stirrer ensures that the bioreactor is evenly mixed and the oxygen is available throughout. Stirring bioreactors are the most often utilized. So option A is your answer. 37. Which of the following is a restriction endonuclease? Hence 2 protease, DNase 1, RNase. So simple, your answer is B, option A, Hind 2. Hind 2 was the first restriction endonuclease discovered. A is the answer. Question number 38. टैक पॉलीमरेज एंजाइम इज ऑब्टेन्ड फ्रॉम, ये भी सिंपल क्वेश्चन है, पहला ही आंसर सही है, थर्मोस एक्विटिकस. ओके, explanation. A thermostable DNA polymerase, that is TAC polymerase, is obtained from Thermus aquaticus, which is a bacterium that remains active under high temperature driven denaturation of double stranded DNA. Question number 39. Which of the following is not a feature of the plasmid? Independent replication, circular structure, transferable, single stranded. Not a feature पूछा है. Okay, so here the answer will be single stranded, option D. Plasmid is a self-replicating double circular extra chromosomal DNA that replicates independently. So plasmid is not the feature of single stranded, it is a double stranded structure. Question number 40. The DNA molecule to which the gene of interest is integrated for cloning is called vector, template, carrier, transformer. So here the answer will be option A, vector. Vector are DNA molecules that can carry foreign DNA, that is gene of interest, and multiply inside host cell. Plasmids, bacteriophages, cosmids, phagmids, YAC are the some names of the particular vectors. So A is the answer. Question number 41. The cutting of DNA at specific locations become possible with the discovery of. The cutting of DNA at a specific location become possible with the discovery of probe, selectable marker, ligase, restriction enzyme. So here the answer is option number D, restriction enzyme. Okay, explanation. Two enzymes that limit the proliferation of bacteriophage in bacteria were found in 1963. The second enzyme recognizes specific DNA sequence and cut the DNA molecule at a specific location. So D option is the correct answer. Next question 42. The introduction of tDNA into plant involves. Option A, altering the pH of the soil, then heat shocking the plant. Wrong one. Exposing the plant to cold for a brief period, allowing the plant root to stand in water, infection of the plant by Agrobacterium tumefaciens. D. Explanation. Agrobacterium tumefaciens, a pathogen that affect a variety of dicot plants, may transfer a piece of tDNA into plant cell. So D is the answer. Question number 43. Which vector can clone only a small fragment of DNA? Cosmids, BAC, bacterial artificial chromosome, YAC, yeast artificial chromosome, and plasmid. So here the answer will be option D, plasmid. Plasmid can have one or two copies per cell. Plasmid is a DNA cloning vector that only clones a short segment of DNA. Plasmids are circular. Extra chromosomal DNA replicates on their own. They are the most often used end strand cloning vectors. The PBR322 plasmid was one of the first widely used cloning vector. So D option is the correct answer. Question number 44. The colonies of restriction recombinant bacteria appear white in contrast to blue colonies of non-recombinant bacteria because of. The colonies of recombinant bacteria appear white in contrast to blue colonies of non-recombinant bacteria because of. Option inactivation of glycosidase enzyme in recombinant bacteria. Wrong one. Non-recombinant bacteria containing beta galactosidase, ये भी गलत है. Insertional inactivation of alpha galactosidase in non-recombinant bacteria. Option D. Insertional inactivation of beta galactosidase in recombinant bacteria. D option is the correct one. Explanation. In recombinant bacteria, beta galactosidase is inactivated due to insertional inactivation and it would not convert the chromogenic substrate to blue colored molecule. It will produce white colored colonies and aid in the selection of recombinants from the non-recombinant bacteria. Now the last question of this chapter. Which of the following is not correctly matched for the organism and its cell wall degrading enzymes? Not correctly matched. Okay, पहला फंगस काइटिनेज, बैक्टीरिया ये करेक्ट है, बैक्टीरिया लाइसोजाइम्स, प्लांट सेल सेलुलेज, algae मिथाइलेज. D is your answer. Okay, the cell wall of algae is composed of cellulose which is degraded by cellulase, not methylase. So D option is the correct answer. And with this we have completed a total of 45 questions of this chapter. I hope this video will helpful to you for preparation of NEET exam. So in my next video I will bring a new chapter. Till then, thank you. Jai Jagannath.